What three elements make up carbohydrates? Well, the answer is right there in the name. Carbo would be carbon. Hydro would be hydrogen. And 8 means oxygen is in it. So carbohydrate, carbon, hydrogen, and oxygen. Macromolecules. We've talked about 4 macromolecules. Those are carbohydrates, lipids, proteins, and nucleic acids. The main function of carbohydrates, if you remember, that's our sugars and starches, and their main function is that quick burst of energy. Think about carb loading before an athletic event. Quick burst of energy. Lipids are stored energy. Fats and oils and waxes and energy is stored there, stored up energy. And also important for insulation and protection in the body. Then we have proteins. Well, the main function of proteins that we talked about was enzymes. Enzymes are proteins, for the most part. Enzymes are proteins. And also they help build body structures. That's proteins. Nucleic acids. We're going to think DNA, deoxyribonucleic acid, RNA, ribonucleic acid. So DNA and RNA, they store hereditary information. Hereditary information. So what are proteins made of? We said several times the building blocks of proteins are amino acids. So amino acids are the building blocks of proteins. Remember, they have the amine group that was NH2. They have the carboxyl group, the COOH. And then they have the R group that changes up depending on which amino acid it is. And there are only 20 known amino acids that all the proteins that we know of. Our proteins, the oak tree's proteins, ladybug's proteins, all those proteins are made from combinations of those same 20 amino acids. The chemical formula of water is H2O. What does that mean? Well, it means that there are 2 atoms of hydrogen and 1 atom of oxygen. 2 atoms of hydrogen, 1 atom of oxygen, and a molecule of H2O, water. In the water molecule, shared electrons spend more time around the oxygen atom than the hydrogen atom. This makes the oxygen atom have what kind of charge? Well, we said electrons are negative. We said if you hang out with negative folks too much, then you end up being negative too. So don't hang out around negative folks. So this would give the oxygen a slight negative charge. It would also give the hydrogens a positive charge because it's pulling that negative away from them. And that's why we call water a polar molecule. It has a positive pole and a negative pole, much like a magnet. That allows it to be the universal solvent. It is able to pull apart, especially other charged particles like sodium chloride. We do that on the board a few times. So it gives the hydrogen a positive charge, and the oxygen has a negative charge. The covalent bond between those oxygens and those hydrogens is sharing of electrons. So a covalent bond is formed by the sharing of electrons. They share it unevenly, so they have a polar molecule. So how are enzymes important in the proper functioning of the body? Enzymes are going to lower the activation rate. Enzymes lower the activation rate the activation energy needed for the reactions in our body. Allowing those reactions to occur fast enough for us to maintain homeostasis. Okay. We have billions of chemical reactions happening in our body right now, and they would not occur, meaning they would not occur fast enough for our body to be able to maintain homeostasis if it were not for the enzymes. Remember, homeostasis is maintaining those stable conditions that are optimal for life. So very important that those are happening. 3 things that affect enzyme activity, well, those were pH. They have an optimum pH. We see a lot of them in our body. The optimum pH is around 7 because we're mostly water, and our blood also has a pH of 7.1. But then in our stomach, we have acids that are digesting our food, so it's a much more acidic environment. So there are enzymes in our stomach that have a much lower optimum pH. So pH affects, temperature is going to affect enzymes. Of course, for the body, our human body, optimum temperature is around 98.6. And what happens if the temperature gets too high? Well, then it's going to denature those enzymes. Often Pat goes, whoop, 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 and changes shape. Now that's going to denature those enzymes. They're going to change shape and not be able to bind their substrate. Okay, so pH, temperature, and substrate enzyme concentration. You want them as close to equal as possible. The closer they are to being equal, then the faster the reaction is going to occur. So substrate enzyme concentration. Enzyme substrate complex are held together by, well, it's between the molecules. We call them intermolecular forces. Intermolecular forces. It holds the substrate in the enzyme until the reaction is complete, and then the enzyme is released as a new product. The substrate is released as a new product. The enzyme does not change in a reaction. It does not change. The enzyme is not used up. The enzyme stays there unchanged so it can do its thing over and over again. It does not use up the enzyme. It does not change the enzyme. The substrate comes off as a new product. A reactant binds to an enzyme at a region known as, well, where they're sticking together there, that's the happening place, known as the active site. True or false? An enzyme speeds up a reaction by lowering the activation energy. Well, we just said that that was true. Lowers that activation energy. If we see it on a graph, you're going to see that without the enzyme, the graph goes up, 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 up for a while before the reaction has enough energy to occur, and then the enzyme, when you introduce the enzyme, then you're going to see the curve go up a little bit, and then the reaction is going to occur at a lower rate than the one without the enzyme. Okay. So which of the following best describes an enzyme? Well, enzymes are classified as organic catalysts. Organic means they're carbon-based from living things. Catalyst means that they're going to speed up the reaction. So the organic catalyst. They are not consumed in the reaction. Sometimes they might have a coenzyme, but not always. And again, sometimes they have a coenzyme, so they don't always work alone. Okay. So identify the reactants. Well, the reactants are the ones that are going into the reaction. So that's going to be our Ca and our Cl2. For the sake of time, I'm just going to go ahead and click that too. So our Ca and our Cl2 are our reactants. Our product, the arrow points to the product. Both start with P, points to the product. So our product is CaCl2. How many atoms of calcium are in the reactant? So calcium's right here. There's just 1 in that group, but then we had that 1 times 4, so there are 4 calciums. How many calciums are in the product? Well, the arrow points to the product. Again, there's just 1 in our molecule times 4 molecules, so that's 4 again. That's good. That means it's balanced. How many atoms of chlorine in the reactant? So we have Cl2. We have CO2 times 4, so that would be 8. And over here we have a Cl2, so there's 2 Cls in the molecule times 4 molecules. So that one will also be an 8. Organization in the body from simplest to most complex. And the smallest thing that has the characteristics of being alive is cell. A group of cells work together to make tissue. A group of tissues working together for a similar function make an organ. A group of organs working together for a function would be an organ system. And then our organ systems together make up our multi-system organism. True or false? When enzymes are finished with their role in a reaction, they cannot be used again. Well, that is false. You can use the enzyme over and over again. It does not change. It is not used up. So be able to identify the macromolecules by their picture and shape. Go back and look at that visual quiz there. Know how to read a food label. You should especially be able to identify lipids. Remember, if it says lipids, it's going to be talking about the fats. Carbohydrates is the sugars and the starches, and I'll usually also have a list of the total carbohydrates, and then proteins, and I'll leave that right there for you. And there's a long chain. We're going to have to be able to identify those macromolecules. So this has our fatty acid backbone, and then it has carbon chains coming off of it. This is glycerol. So we're familiar with glycerol. And its structure looks like a ladder. Glycerol looks like a ladder. For the sake of not trying to draw it and using up a lot of time, we're just going to talk about this. Carbohydrate is a monosaccharide. Remember, mono means 1. Okay. So that's our single sugar. They're usually like 5 carbon rings or 6 carbon rings, so it's going to be like a little pentagon shape or a hexagon shape. Disaccharide is going to be 2 of those monosaccharides bound together. Polysaccharide is going to be 3 or more. And we looked at some pictures of those, so make sure you're familiar with those. Remember, our nucleic acids have 3 parts. And we'll have like an NH2, if it's at a amine group, NH2. And then it's going to have the carbon in the middle that's connected to the R group down here. And then it's going to have the COOH to the side. That is a carboxyl group. So we're going to have N, H, H, and the C, and our R group that changes depending on the amino acids. And then we have a COOH, carboxyl group. Make sure you're familiar with how those look as well as what each of those groups look like. So if it says something like how many oxygens are in a carboxyl group, well, you're going to say 2 oxygens because it's COOH. Okay. And then you need to be able to balance chemical equations. So we practice those a good bit, and we'll leave these here for you to practice as well. Remember, you start by listing what you have. You would list your N and your H down under. You'll want to work these out on a bigger piece of paper, and you can go back in and put your coefficients in there. I hope that helps. Remember to study. Look back over. Look over those 2 questions that I said be really aware of on the Amoeba Sisters handout that we did. Over enzymes, 2 questions there that you particularly needed to pay attention to. All of this that we've just talked about on here. And good luck on the test. Don't forget to study.